Good morning, everyone. Welcome to the Malaysian Society for Molecular Biology and Biotechnology MSMBB Seminar Series. The MSMBB Seminar Series is co-organized by MSMBB and the Department of Parasitology in the Faculty of Medicine, University of Malaya. This is assisted by the Visibility Unit and PTM, University Malaya. Before we start the seminar series, I'm happy to invite the President of MSMBB, Prof. Dr. Lau Yiling, to give a welcoming speech and to announce our MSMBB STEM TikTok winners. Prof. Lau, I pass the microphone to you. Prof. Lau?
Sorry, can you all hear me?
Yes.
Okay, let me... Is my slide showing?
Not yet, Prof. We can see it now.
Okay, can you hear me?
Yes, Prof.
Okay, everyone. Welcome, everyone. It is my great pleasure to welcome you to the Malaysian Society for Molecular Biology and Biotechnology Seminar Series with the aim of promoting scientific advancement by sharing knowledge in the field of molecular biology and biotechnology. This year, the seminar series is co-organized by Department of Parasitology, University of Malaya, with the help of Visibility Unit and Pusat Teknologi Maklumat, Two seminars will be held concurrently using Zoom platforms. Before we begin, I would also like to introduce a few initiatives that have taken place this year and next by MSMBB. Well, MSMBB has successfully organized the MSMBB STEM TikTok video challenge last month to create public interest in science, technology, engineering, and mathematics STEM through social media. Each team was expected to shoot a 1-minute video in English through the TikTok application by using the MSMBB backdrops. I'm proud to officially announce the winners today. The best content goes to Cham Mui Hui, University Putra, Malaysia, with the title Scopage And team members, No Akila, No Hira binti Afidi, and No Hana Nabila binti Nabizas. Congrats. Another best content goes to No Amariza Akmal binti Muhammad Kamal, University Putra Malaysia, UPM, with the title Spray and Get Protected. The most likes goes to Adam Daniel binti Arifin, University Putra Malaysia, with the title Chronic Wasting Disease. Another most likes goes to Sharon Tiang Chin Yi from UCSI University with title Eyes Wide Shut. Team members Wong Pui Man, Yo Jia Hao, LV Jamima. Yeah, congrats to the winners. I will also take this opportunity to thank the judges, Dr. Kanti, Jeremy, Dr. Amir, Garia Ang, to spend their time in judging the competitions. Of course, it is not to forget also the organizer, Associate Professor Dr. Siti Sarah Oman from UPM, Dr. Crystal Lim from UCSI, and also helped by Associate Professor Dr. Wang Xiok Mui. Thank you to all of you. Next, I would like to introduce a journal run by MSMBB, Asian Pacific Journal of Molecular Biology and Biotechnology. It is a Scopus Index journal. Feel free to submit your articles to this email address. We also run an EduSTEM grant, which support the projects working with virtual science, virtual STEM, which is science, technology, engineering, and mathematics educations. The deadline is 31st of December 2020. Feel free to submit your grant applications to MSMBB website or email. I also would like to announce our conference, which will be run next year, 1st to 3rd of June 2021. It is a 4th International Conference on Molecular Biology and Biotechnology 2021. The abstract submission deadline is on 1st of April 2021. More information can be found on our website, www.msmbb.my. That's all for the announcements. I would like to present to you today's main agenda, which is the MSMBB seminar series. Thanks to Professor Manikam Ravi Chandra and Professor Jennifer N. Harris Krishna for joining us as speakers today. We will be in 2 rooms as mentioned earlier. One is in main room. Professor Ravi's talk will be in main room. We will be moderated by Dr. Amira Amil and co-hosted by Dr. Arun. Another one will be in the breakout rooms. Whoever interested to join Professor Jennifer, please join the breakout room now. And the moderator is Dr. Poon Ching Tan and co-hosted by Freddie. I will pass the floor to the moderators now. Thank you.
Thank you very much, Prof. Lau, and congratulations to all winners. So I hope all participants can enter the respective breakout room. So you'll be able to see the breakout room icon at the bottom of your screen. And if you're going to listen to Prof. Jennifer's talk, kindly enter the breakout room. For those of you who wants to listen to Prof. Ravi Chandran talk, please remain in this room. We will start soon. Good morning again, everyone. I am Amira Amir, and I will be your moderator for today. Just a gentle reminder, this is a 45-minute talk followed by 10 minutes Q&A session. Kindly post your question in the chat box, and I will read out the questions to the speaker after his talk finishes. Our speaker for this session is Prof. M. Ravichandran. He is the Dean of Faculty of Applied Sciences, Ames University. He obtained his MSc in Medical Microbiology from Christian Medical College, Vellore, and gained his PhD in Biotechnology degree from Anna University, Chennai, India. Currently, His research interests are genetically engineered cholera vaccine and formulation, molecular diagnostics, and phage therapy. He has 76 publications in international journals, supervised 40 postgraduate students, filed 8 patents, and commercialized 3 products on diagnostics. He has been awarded 44 national and international awards for the academic and research excellence. He was an associate member and the committee member on Top Research Scientist Malaysia of the Academy of Sciences Malaysia. He was the former Vice Chancellor of AIMS University Malaysia. Without further ado, I would like to invite Prof. Ravichandran to give his talk. Prof, the floor is now yours.
Good morning to everyone. Thank you, Dr. Amira and all the team members, Prof. Kantimati and Prof. Tang and all the MSMBB members. Hope you all can hear me. Just say yes or no, so we know that.
Yes, we can hear you, Prof.
What I'm talking is audible. Because we are in a challenging time. Like talking in front of a computer, and nice to see August audience, and very enthusiastically you have participated, and MSMBB has been doing an excellent job, and congratulations and thank you for the opportunity. Without further ado, I will share my slides. And can you see my slide now?
Yes, Prof.
Okay, thank you. At least one person give a response in time being, then we know that we are live. Very good morning to all of you. I'm just, today's presentation is on a bacterial vector vaccine platform for emerging diseases. So I'm going to share my experience on developing a bacterial vector vaccine for cholera. So I'm just wrap it up with some principle behind the vector vaccine principle in this talk. So my content of this lecture will be on vaccines. I just give a very brief overview on different types of vaccine, advantages and disadvantages, and what are the barriers of DNA vaccine, and what is a bacterial vector, and how a bacterial vector can efficiently deliver a DNA vaccine, just a mechanism, immunological mechanism. And then I will start with the journey of cholera vaccine development, or how we developed it. I just share with you some of the experiences, and how as a scientist, sometimes we plan a strategy, we have to change back, and then we move on, and how research is sustained for the last 20 years. I'm still alive, and the vaccine has been progressing, and various generations of students have been working on it. And I can say nearly more than 50 over students and staffs have been working on this development, and many funding agencies has generously contributed for this vector development. And I'll just tell you how we broke the cold chain barrier, because one of the important thing on vaccine is that it has to be maintained in a cold chain. And so I'll just summarize with the platform and its potential to be used for emerging diseases. So as a vaccine, we know that vaccine used to stimulate the production of antibodies, and it provides immunity against one or several diseases that is prepared from the etiological agent, and normally it is produced from its antigens or gene, or that should not cause a disease but should elicit the immune response for the vaccine. And we have seen in the textbook that we have different types of vaccines. We have whole cell vaccines. It can be a live bacteria live virus, or a killed bacteria kill virus, or a subunit vaccine, and it could be a peptide or recombinant, like for example, hepatitis, and so on. So that could be a different type. And newer vaccines such as Currently, you can see that RNA vaccine or a DNA vaccine are used. So this is a common picture we see in the current COVID era. There are so many different type of vaccines are there, and it includes the attenuated, inactivated, and so on. So I would like to focus on the DNA vaccines. So the DNA vaccines can be delivered as when it came in 1990s. They used to coat that with a gold nanoparticle. Remember the gene gun RITARD used to sell. So they used to coat it with the gold nanoparticle and then shoot it on the muscle, and also injectable naked DNA vaccine. They are delivered intramuscularly, and recently many of the DNA vaccines or RNA vaccines are encapsulated with the bilipid membrane. And today we'll be focusing on how we can use a bacteria to transfer the DNA vaccine vector into the host efficiently, non-invasively, in a cost-effective manner. So this is when we develop a cholera vaccine, these are the principles we pulled from WHO. It should be we should avoid the multiple dose. It should be single, and it should cause less adverse reaction. And there should be no requirement of buffer. It should have long shelf life. The most important is storage at room temperature, and it should be inexpensive. The challenge, more than 1 billion people in the world can't afford. It should be less than $1 a dose. That was the challenge we kept in our mind when we developed this vaccine. So there are so many vaccines people are developing. If it is going to cost $20 a dose or 2 doses, $40, I don't know how many people can afford in this planet. So we have to use the science in an efficient manner so that we can protect every civilian in this planet. So just brief principle about DNA vaccine. They actually are plasmid. It's a normal plasmid, and it should be encoding a gene, and it will have 2 promoters. It has 2 parts. It has the prokaryotic part. It's a bacterial promoter and all the necessary elements, and it also should have a prokaryotic promoter. And so we used to call in molecular biology, we call it a subtle vector. So it can be propagated in E. coli, and it can be delivered into mammalian cell. So it basically the gene is transcribed inside the muscle cell, and it translates the protein into a protein, and that antigen is delivered inside the cell, and it will elicit the immune response. So these DNA vaccines are basically, as I mentioned earlier, it can be delivered in these 3 different modes, and in this talk we'll be focusing on the plasmid that will be delivered using a bacterial vector, the theoretical aspect of it. And coming to the DNA vaccine, when you want to develop a DNA vaccines, you have to make sure that it does not have an antibiotic. As much as possible, the antibiotic should be there. But ampicillin should not be used because people have allergic reaction to ampicillin. So you have to make sure that the DNA vaccine vector should not have an antibiotic resistant gene, ampicillin resistant gene, because in the production process you'll be using ampicillin. If the trace element is there, then that might cause some allergic reaction. So kanamycin may be a good target. Similarly, you have to minimize the non-essential genes because most of our people who work in molecular biology, PUC18, PUC19, PBR322, most of the vectors are derived from that, and they have some of the unnecessary genes are still there. So you have to make sure that only the necessary elements are there in the DNA vaccine vector, and mobilizable elements are not allowed. So if you use a mobilizable, it might transfer into the another host. So all this precaution has to be done when we design DNA vaccine vector. And there are so many DNA vaccine has been developed, but I don't see anything in the market right now. And some are in clinical trials, and for HIV, influenza, TB, diabetes, and cancer. And these vaccines, DNA vaccines, are very good in inducing immune response and easy to produce. It's like just plug and play. We can just put the gene inside and use it. And it also, DNA is quite stable, and it's low cost, but in terms of immunogenicity, there is always a challenge there. And in large animals, the efficiency is a little less, and definitely you need a multiple doses. And there is no efficient delivery. Most of the tools are invasive. You have to inject or other methods. So these are the major barrier in DNA vaccines. The currently developed the Moderna vaccine also is RNA. RNA, we know any researcher who works on RNA, they know how to maintain an RNA. You need minus 70 degrees centigrade, and it degrades very fast. And if you do an in vitro transcription, you might get 1 microgram, 2 microgram using a T7 RNA polymerase or MMLV RNA polymerase. You want to inject 100 microgram per person. I don't know how you make it, but there are technologies we can use to synthesize them artificially. So the barriers is you have to use a gene gun or an injection or sometimes nebulizer and so on. I think these are ineffective and painful and difficult to administer. Orally, you can give a DNA vaccine, but our gut pH will chew it away and degrade it, and it will be easily eliminated by the system. So the best model, I believe, is the oral delivery through a bacteria will be the best way. It will be low cost and easy to administer, single dose, and it's self-replicating, long-lasting immunity. And if you look at our gut mucosa, and the surface area is quite good. And our mucosa is our first-line defense. Even in the mouth or respiratory tract or in the gut, malt or other associated tissues, mostly the IgE antibody, the first line of defense. Any pathogen or virus comes, it will be able to neutralize through the IgE secretory antibody. So I just want to tell what are bacterial vectors. Bacterial vector basically is a bacteria. It's a live attenuated bacteria, and they are very good in mucosal delivery of the antigen. It can be administered orally or intranasally, and I'm not focusing on other routes. And both the mucosal and systemic immune response it can, because when it goes to the gut, the M cells can uptake the bacteria, so it can cause an intracellular presentation, and it can elicit the cellular immune response. When it delivers in the mucosa, it can induce a mucosal immunity. Many bacteria have been used, like Listeria, Salmonella, the spelling is Typhimurium, and Vibrio cholera has been used, and Lactobacillus has been used as a delivery of these DNA vaccine plasmids. What happens is when they go into the bacteria, once it's uptake by the cell, the lysosome breaks and releases the DNA vaccine plasmid inside the cell that will express it uses the CME, cytomegalovirus promoter. It's a eukaryotic promoter, so that will transcribe the gene which is put in the plasmid, and the antigen is expressed intracellularly. And the DNA plasmids are released in the cell and then taken up by the dendritic cells. This is one example how when it is taken up by the dendritic cells, the antigen gene is expressed as a protein inside the cell, and the gene presented on MHC class 1 or class 2 molecules, either through CD4 or CD8, and the whole immune reaction can happen. It can both go to the B cell arm or T cell arm. Whatever gene you put, the antigenic response will be developed through these immune cells. This is another cartoon about how a bacterial vector can be used to vaccinate a pathogen. So basically, you have this bacterial vector. So the gene is cloned, and it can put inside a bacterial vector. So it can express the gene in two different ways, and one, you can put it chromosomally, so it will express the antigen on the chromosome. Extracellularly, it will express the gene, and it can also integrate into the genome, but that might not work for DNA vaccine plasmid. So one of the essential elements, you must have a efficient eukaryotic promoter. So it can express the antigen intracellularly. Sometimes it can secrete the protein into the cell. And as I mentioned earlier, the humoral immune response or cell-mediated immune response can elicit, and then it can give the protective immunity against the target. So I'm just switching gear now. So we know that there are many different types of vaccines. So DNA vaccine is one of the easiest way to develop a vaccine. I won't call DNA vaccine, call a nucleic acid vaccine. So the efficient way to develop if you have the emerging pathogens are coming in. So once the sequence are there, we can create a DNA vaccine plasmid. So how we understand that the DNA vaccine delivery, one of the more, I would call a sustainable way to deliver a DNA vaccine could be the bacterial vector. So I'm going to tell a story about how we develop one live bacterial vector. The example here is cholera. So this work, I think so many people have been involved. I say more than 50 over people, researchers and students have worked on this. It started way back in 2000. And the first aim, we want to develop an oxotrop. So we want to develop a bacterial vector, the vaccine for cholera, which is programmed to die by an oxotropy. So we cloned the Heme and M gene of Vibrio cholerae. At that time, the gene sequence, even Vibrio genome was not available in year 2000 and early. So we constructed a mutant of Heme gene, and that's an oxotrop too. I'll tell you what is the principle behind it. So we developed various strains, and we VCUSM2, it had some property. We removed the toxigenic property. We developed another strain, and then there was a problem in colonization. We modified the strain, and currently this strain has been formulated, and it is not toxigenic, and we developed a cold chain formulation, and currently it is undergoing the GLP-ICH guidelines toxicology studies at IFORM. So I'll just let's start with this our principle. Any vaccine, live attenuated vaccine should be live. Live means it gives you better immunogenicity, and it should colonize very well. If it is a cholera vaccine, for example, here it should colonize the gut so that the cells can elicit the mucosal immune response, and it should give protection, and it should be not reactogenic. And then the most importantly, if you want to reach the bottom billion, it should be cold chain free. So this is the statistics of cholera. I think it's a poor man disease, and a global burden of cholera is almost 250,000 deaths every year. And mostly, I think 2017, there's a spike of cholera, and mostly in sub-Saharan Africa. There are at least 69 countries that are affected by this particular disease, and more than 1 billion cases in these countries. And the best way to prevent is good water and sanitization. And so vaccine is one of the way we can use it. That's the reason we have been developing this. And what are the strains? The strains which we have developed are the live oral attenuated vaccine strains, and they are specific to the O139 serotype. And the first strain which we developed was a metabolic oxotrop that requires an extraneous chemical called ALA, aminolevulinic acid, for its survival. Later, we converted into prototrop, so we put back the mutation so it doesn't require the chemical. So first thing, the first prototype when we develop, we want to, as I mentioned to you, we want the cells to program to die. So we mutated the heme gene, that's a housekeeping gene of Vibrio cholerae. It's one of the important genes for its metabolism. If the gene doesn't work, the bacteria will not grow without the supply of extraneous aminolevinic acid. So the study was focusing on mutating this glutamyl tRNA reductase. That is actually heme gene of Vibrio cholerae. Nothing to do with the heme metabolism. It is glutamyl tRNA reductase involved in aminolevinic acid metabolism. So we published in Vaccine in 2006. This just a summary. This is the pathway. We studied the pathway. We know this heme gene that encodes glutamyl tRNA reductase is a rate limiting step. So we knocked out this gene. First, this gene was not available. So what we did is we took the gene from E. coli. This gene was available in E. coli, so Vibrio genome was not available. So based on the similarity, and we designed some primer, and we did an alignment. We look at the conserved region, and we pulled out that particular fragment, and we cloned and mutated this gene. This is the gene which we obtained. So when we discovered, we got two genes. One is a heme and heme gene. Heme gene was used for vaccine development, and heme gene now currently tagged as LOLB gene, LOLB gene, and it has been this sequence is very, very specific for Vibrio cholerae species. So we developed a few diagnostic tests based on this, and also biosensor for detecting Vibrio cholerae, and we have few paper on heme gene. And heme gene we have used to develop an oxotrop. So what we did, just a brief overview, and anyone interested in genetic engineering, now called genome engineering, you can do it. You can understand it's a long process. So basically, we clone this gene in a vector, and one background I have to tell you is that Vibrio cholerae is not E. coli. It has a membrane-bound DNAs on its membrane. So when you transform anything, it will chew up. So we have to transfer the gene only by conjugation. So by normal method, it does, but the efficiency is very, very low. We can even do by electroporation, but the frequency is very low. So we took this and put it in we clone it in a vector, and then we did a insertional mutation by putting a kanamycin gene capsid in the heme gene, and then transfer into a mobilizable suicide vector. And this particular vector is called PWM91, and it has a gene called SACB gene. It codes for saccharase. And what happens is when this plasmid is transformed into this particular, first of all, it's transferred into E. coli. It grows very well in E. coli, no problem. So it will be by filter mating method, this plasmid was transferred into Vibrio cholerae. So once it goes into Vibrio cholerae, the replication, R6K replication in this particular plasmid won't work in Vibrio. So it becomes a suicide plasmid. So this plasmid get integrated into the genome of Vibrio cholerae. And this gene, there's a gene called SACB gene, which is toxic to Vibrio gram-negative bacteria, Vibrio cholerae, when you grow them in the presence of sucrose. So when we do when we grow them in the presence of sucrose, and the recombination happens, and the whole fragment, the plasmid backbone is lost, and there will be few stages of recombination happen, and then we screen for the mutant. This is just the overview of the strategy, how the genome engineering. Now the new word they coined is called genome engineering. There are a lot of other new plasmids come now to do to fasten the construction of the strain.
Hello? Yes, ma'am. Yeah, சொல்லுங்க ma'am.
So basically the environmental safety features, how we intended was, when you grow bacteria, this is a wild type I've pictorially shown here, and this is the mutant. So the strain which we have developed is a mutant for aminolevulinic acid, ALA. So when the bacteria grows, so what happens is that it divides by binary fission. The cytoplasm exactly divide by 50%. So when the strain was grown in the presence of ALA, every division the ALA get depleted. So what happens is at certain stage there is no more ALA. So the bacteria, when it is given to a animal model, so it grows for a few generations and dies by itself. Okay, so that is the principle of our auxotropy, and but we left all the cholera toxin, every gene intact in the strain. So we developed the strain. We found that the ALA level went down, and the bacteria growth eventually depleted after some time. So that was with every multiplication, the cellular ALA is depleted, and it failed to multiply, but wild type grows continuously. That was a principle. We did the challenge study in a RITARD model. RITARD is rabbit intestinal tiled adult rabbit model. So it is the only model available for cholera. We have to manipulate the gut of the rabbit to create this. We did a challenge study. So all the rabbit vaccinated with our vaccine strain survived, and unvaccinated rabbit died, and it worked very well in terms of protection. It was 100% protection when we studied in the animal model. And we were a bit worried about the safety of the strain. We don't want to release the bacteria. Strain should not survive in the environment. So I don't know why I did the study. And that time there was an outbreak of, in year 2000 in Kelantan, there was an outbreak of cholera. So we thought we should study in the sea water and river water. We studied, we put this bacteria and then see how long they survive. And this vaccine strain could not survive longer, but the wild type was able to be persistent in the river water and sea water. And so this is the overview of the result. It colonized and multiplied in the small intestine, which looked for all the antibody, antitoxin, IgA, IgG, and vibrio-sural antibody, which is one of the hallmark of the immune system, immunity for cholera. And it had reactogenicity in the illeal loop. When you inject an illeal loop, it's a fluid, but fluid was there. And it was 100% protective, and it was environmentally safe. So the bottom line of our this particular construct is everything is fine, it is protective, but it has a reactogenicity was there. So we need to remove the reactogenicity. So the reactogenicity is due to the virulent factor. Any bacteria, the reactogenic means virulent factor. The major virulent factor of cholera is cholera toxin A, ACE, and ZOT. So these are the virulent factor we want to do, but we don't want to talk when we started this, we want to keep all the virulent antigen repertoire intact so that the body can develop a good immunity. So ACE and ZOT, no other choice, we deleted it in the next strain. And CTXA we mutated. I'll just tell about it. So we filed a patent on it, and we filed a Malaysian patent and US patent, which is still valid. We also filed an European patent and so on. So the bottom line of this particular strain is it's a good colonizer. It colonizes the small intestine, and it gives a very good antibody titer. But it is mildly reactogenic in animal model due to the presence of the CTXA gene. So if go back to our principle of this vaccine, it is cold chain free, and it is live attenuated. It's a good colonizer, and it gives good protection. And but it was still reactogenic, So basically due to the auxotropy, but the CTX operon is still intact, the cholera toxin is still intact. So we did the next stage of development. It was done by one of my postgraduate students, Ms. Melissa Chan. And we did a site-directed mutagenetics of the CTXA. So we just mutated only the active site of the particular cholera toxin. and so that we can mimic the natural infection. And this one was mutated on this assa and ZOT has been deleted, and we did by genome site-specific recombination, we transferred those gene. The CTXA gene was it had 2 active sites, and if you look at the cholera toxin, it's a pentamer. Okay, it has 5 CTB subunit, which is not toxic, and the CTA is toxic subunits. So basically, we mutated those CTXA gene and also deleted the ACE and ZOT gene. And there are 2 amino acids, active sites is public knowledge. So these are the 2 amino acid codon which we mutated by side-directed mutagenesis on the plasmid, not on the host. So after that, we transfer back into the host. So we did illeal loop studies, and we were very happy to see that, for example, you can see that loop 0139, the wild type, causes a lot of fluid accumulation, but this vaccine strain absolutely did not cause any fluid. We was very surprised. The toxin gene is there, only the 2 amino acid has been changed in that. And it is there, and we have been monitoring this particular strain for so many generations. It has not reverted. There is no reversion of the mutation, and it still produces the antigenic part of the cholera toxin, and the fluid accumulation was also almost 0 in 14. So we also did this quantitate that the cholera toxin by ELISA, and we found it did produce. We had so many derivative of the strain. We have single copy, double copy cholera toxin mutant, and the cholera toxin amount is there between around 1 to 5 nanogram. We are able to observe by ELISA method in this strain, but these toxins were not toxigenic but antigenic. And we also did an infant mouse colonization. That's very important, because when you produce a vaccine, it should stick to the gut epithelial cells. So we are able to find they were able to colonize almost equivalent to the wild type. So 2 into 10 to the power 4, and wild type was 2.2 into 10 to the power 6. So we are able to get, at least to some extent, they were able to colonize the intestine. So this particular strain was very good, absolutely not reactogenic. And they produce less CT compared to the parental. The cholera toxin production was less, and the colonization was compromised in this particular strain due to its oxotrophy nature. So conclusion, so the mutation was successful. The strain was not reactogenic, but it did not colonize very well. So the colonization property was removed, and it was live, non-reactogenic. Gave good protection, but these 2 properties compromise the strain due to the presence of oxotropy. So now we have to revert back the first experiment we did. So we mutated the heme. Now we have to put back the heme gene. So that's what we did in the next study. So we developed a prototype of this version of this strain. So what we did, basically the same strategy. We put back the heme gene into it. And we found, we are so excited to see that it have a very good colonization, almost roughly equivalent to the wild type strain, and it also produced a good amount of anti-cholera. It produced a good amount of cholera toxin, which is antigenic but not toxigenic, and so the CT production was good. And the fluid accumulation in the intestinal loop, which I told you, the illeal loop assay. So absolutely no reactogenicity of this particular 14P strain. And we also found good amount of antibody against the cholera toxin and CTB and CTA and so on. I just want to pass here and tell that CTB is one of the immunological adjuvant. If you search PubMed, and CTB has been proven as one of the great immunological adjuvants. So we that antibody that is also expressing very well in this particular strain. And fluid accumulation in the illeal loop assay also was tested. And also the vibricidal antibody. Vibricidal antibodies, the antibodies that can kill the Vibrio in the presence of complement. The titer also was quite high titer we received this strain. And the challenge studies, we are able to see that it gave a protection in the rabbit model. So basically the ALA prototype carrying the mutated CTX operon was successfully constructed, and the prototype strain was able to grow without no need for ALA now. So it cut down the cost of production, and it colonized very well. And there is no pathological changes. We also did the histopathology. We have published 2 papers on that. There's no histopathological changes when we use this strain in the animal model. And it releases a good amount of antibodies, and it protected the rabbit in intestinal studies. So, so basically we have a strain, everything is achieved good. It is a good colonizer, live attenuated, and it gave 100% protection, and it was not reactogenic, but the cold chain is still was a problem. And the time I had collaborated, I had a colleague, Dr. Prabhakaran. He has around 20 or 30 years experience in industries. So he developed a formulation that can keep the bacteria at room temperature for 140 days. So, so that part was, the 2 studies has been done. We did a repeated dose toxicity evaluation of our formulation in rat model, and we found the absolutely non-toxic, and this was published in Vaccine. So the same study now we are doing in a GLP facility to prove the toxicity, to do the repeat the toxicity, then we can develop, go to the next level. And this formulation also has been published, how can stay outside the cold chain for 140 days. So we published, Dr. Prabha team has published in BMC Immunology. So we have a bacterial vector, that's a cholera vaccine strain, which can stay at room temperature for 140 days. So, so I'm glad to say that we broke the cold chain. And you know, if you see any vaccine development, the transportation cost of a vaccine is almost 25%. Imagine the current vaccine which you want to make, that is 4 degrees, you know. If you want to maintain at minus 70 degrees in your cold chain, I don't think it should be much more than 23%, 25%. So we have this was done in collaboration. The strain was developed at USM and then improved at Ames University, and the formulation was done at Ames University. So it's a good example of a collaboration, and we have a low-cost cold chain-free vaccine, bacterial vector we have. And I'm glad to say that our strain has been, in one of the presentation I pulled out that our strain is in the preclinical stage according to the WHO. And so we have achieved a good strain, live attenuated, good colonizer, cold chain free, non-reactogenic, and gave a good protection. So we are still progressing toward developing this strain for cholera. So we are the toxicology studies from 2000 and 2019, and now the toxicology studies are going on. And we also have a trademark of this vaccine has been registered as a MyCall. And we have so many patents been backed for this this particular strain. So I just want to tell the scientists and students and researchers, this is a good example of perseverance and sustained research for the last 20 years. The strains are still alive. It has been deposited in the public repository. It means as per the patent requirement, we have deposited in the repository. So the research is sustained and continued, and some of the students who are working on this might not know me. There are so many generations, have many grandchildren students working on it. And so there are a few generations of scientists developed this. So basically, we found this this particular strategy can be used for developing. If you have a DNA vaccine, it can be it's only theoretical. It can be transformed into this strain, and we can develop any emerging vaccine in a faster phase. And this is just a nutshell. This particular platform has been protected by various patents and trademark, and we have a series of publications from vaccine and BMC immunology and so on. So we believe that this this particular platform can be used for any emerging diseases in a faster way. So in nutshell, these properties of this particular platform which we have developed, in addition to the cholera, this one is oral formulation, easy to administer, is cold chain free for 140 days, and it is live. And it has a built-in safety property because that it has CTB, the cholera toxin B, which is known to mucosal adjuvant. And it is it also the cholera toxin, the B subunit and A subunit is 85% similar to E-Tech cholera. So it also can be used as a E-Tech. We have one project going on currently doing it to check it efficacy against E-Tech, and preliminary studies are promising, and it has a great market potential as it is cold chain free. There are so many people are involved in this. I think it was the seed was planted by Professor Zainul F. Zainuddin. He's no longer with us, and he was the man who got the idea to do the oxotrophy strain, and so many people under this. And currently, all the current students are not all pictures are there in this, and it has been rejuvenated at this pandemic. And now currently working with Professor Nurazmi at USM. He's a well-known vaccinologist. So we are taking this vaccine to the next level as a potential bacterial vector DNA vaccine vehicle. So with this, I'd like to thank you all. This is the campus I stay in. This is Ames University in Kedah. I stay in this third house in this beautiful campus, which is located in the Pujang Valley of Kedah. Thank you very much for your attention. I'll be happy to answer any of your queries.
Thank you very much, Prof. Ravichandran, for a very insightful talk. It's obvious that you've put in a lot of work in your study. Right, we have a few questions from our participants. Are you ready, Prof. Ravichandran, to answer them?
Yeah. No
right.
problem.
So our first question is from Prof. Lau, and Dr. Roslina Jawan also shares the same question. So they're asking about the new COVID-19 vaccine. Is it made from plasmid vaccine or subunit vaccine? And do you think it will be effective?
I think it's a very sensitive question. The new COVID vaccine that there are so many people develop. One is Moderna, and other one is Pfizer, Intech. They are RNA vaccine. And if you look at my slide, I showed you the encapsulated DNA. It's encapsulated with lipid membrane. That is the science behind it. That is, it's not DNA vaccine, it is an RNA. And the RNA, the mechanism is they have to inject 100 microgram. I think one of the vaccine was 100 microgram. I was wondering how they're going to make it. I don't know anyone done in vitro transcription knows making 1 microgram of RNA is like you have to stand upside down. So I think most probably they'll be synthesizing artificially or in an oligosynthesizer, or it's a quite big molecule. So that is the science behind it. It's an RNA molecule encapsulated in a bilipid membrane, and it has to be stored at a low temperature and minus 70 degrees centigrade. Some of them claim that it can stay at 4 degrees for some time. In terms of efficiency, they have done several trials, and they claim that it gives 90 to 95% protection. in volunteers in a blinded study. So when they unblinded, they found that the volunteers injected with this RNA vaccine 95%, 90 to 95% protection. But what we know is it is working, but we do not know the long-term effect. This vaccine has not been, if you look at the development, and they have not done any preclinical toxicology studies, I don't see anything. It is actually a fast-tracked, and all the results are based on the previous RNA vaccine, not this particular RNA vaccines. So the long-term effect we do not know. But it is, according to their report, it is not a published report. It's been published in news media only. It's not a scientific report. So based on the preliminary finding, it's impressive. But I do not. But it does not cause any GMO. I think in science, I think we have to promote we have to stop the miscommunication. So it does not cause a GMO effect. It doesn't even enter the nucleus. It doesn't recombine with our DNA. It stays outside in the cytoplasm, and possibly it degrades by itself. The RNA molecule, the life, the self life. We all know that RNA copy number and RNA stability in a cell. The RNA won't stay very long. So it is. I won't call it as a GMO.
Thank you very much, Prof. Ravi, for your thoughts on the COVID-19 vaccine. I think a lot of people are anxious to have it out already.
Yeah.
Right. So we have the next question. It's also from Prof. Lau. So she's asking how WOT improved the colonizer in infant mice of HEMA mutation study.
Say it again.
How does WOT improve the colonizer in infant mice of HEMA mutation study? HEMA mutation study, sorry.
I don't know what is WOT. Improve the colonizer in infant mouse. Okay, I understand what it is. Because HEMA mutation actually, you all know prototrope and auxotrope. Auxotrope are the bacteria that require certain amino acids or chemical for its growth. That is auxotropy. Okay, that's why we all study in molecular biology. Prototropes are the one which can grow very well. Okay, this particular strain, the first strain which you develop, it's an ALA auxotrope. Means you have to supply this this particular chemical ALA, amino levulinic acid, for it to grow. So since this strain, as I mentioned to you, it can only multiply few generations. After that, it dies by itself. So when you give it to the mouse model is basically an infant mouse model we use to see how many bacteria stick onto colonize onto the mouse intestine. Okay. Since the bacteria itself is not able to multiply very well because it has only limited multiplication, so the colonization efficiency of the HEMA mutant was less. So that's why we reverted the mutation in the last strain, and we made a prototype of that.
Thank you, Prof. Our third question is from Dr. Roslina Jawan. She asks, what is the difference between this vaccine that you presented just now with the current vaccine that is available for cholera treatment?
So our vaccine is actually, we focus, the cholera can be caused by two major serogroups, O1 and O139. So ours is focused only for O139. That's number one, that's the difference. Number two, I think this is the only live attenuated and very systematically genetically engineered cholera vaccine in the world, I can say. Most of them, they take the strain which is deficient of cholera toxin or naturally deleted strain and things like that. So in this one, we have systematically mutated. We know the whole genome. We have the sequence of the whole genome, and we know the stability, and it's oral, and it's the only oral cold chain free formulation which can stay at room temperature for 140 days. So that is the beauty of our this platform.
That's quite impressive, Prof. Ravichandran. Our next question is from Dr. Cheong. She says, thank you for the informative talk. What are the advantages of this developing vaccine candidate compared to the currently available cholera vaccine? Probably you've answered this, or you want to add
Yeah,
on?
maybe the currently available vaccines, actually there are two vaccines available. One is called Shancol. That is actually killed cholera vaccine, bivalent cholera vaccine formulation. So that you need 2 doses, and it still needs a cold chain for transportation. The other vaccine is called Dukoral. Dukoral is a live O1 vaccine. That also, again, it needs a cold chain, and it also need double dose. So our vaccine does not require that. It's cold chain free. That is the difference.
There's another question by Mr. Nakib Rafikin. So he's asking, why does some strain they are able to be maintained at room temperature of 4 degrees, while this VCUSM 14 able to stay viable at room temperature of 4 degrees?
Okay, because it's a formulation strategy. So when you want to formulate the bacteria, you have to follow its nature. These bacteria are present normally in river water, sea water. So the formulating chemical you have to use, which is it's actually a proprietary formulation, and we use the nature as the example to formulate the strain. So it stays comfortable, it's viable for 140 days at room temperature.
That's interesting. Our next question is from Dr. Aruchelvan. His question is, can bacterial vectored vaccines be used as anti-cancer vaccine?
Yeah, actually, if you Google, and you can see that in vivo, and there are many companies developing a DNA vaccine for cancer using bacterial vectors. So you can only this is plug and play. You just need to change the antigen. And if anyone working on cancer, you want to use this as a model, we can collaborate. It will deliver much better and safer.
Right. So anyone who wants to collaborate with Prof. Ravi, make sure that you contact him after this talk. Our next question is from Dr. Adeline. She says, thank you for the excellent talk. Is it necessary for the bacteria vaccine to be a good colonizer? If it manages to induce a good immune response but does not colonize, is that okay?
If it doesn't colonize, it won't elicit an immune response, because you need connection to elicit. Let's say if it floats around in the intestine, it will go into the stool, it will pass away. So so it should colonize, then only it can interact with the GALT and MALT, gut associated lymphoid tissues, and mast cell associated lymphoid tissues. So interaction is very important. If anything just goes by and our immune system has no chance, look at it. So it has to colonize.
Right. Thank you, Prof. Ravi, for the clarification. We have a comment from Dr. Sazali who says, excellent talk, Prof. Ravi. Always good to hear you talk.
Thank you,
always
Prof.
good to hear your talk. Yeah.
Thank you, Prof. Sazali. I'm happy that you are there.
There's one last question from Hannah. What is the survival rate of this strain in storage?
Yeah, it's basically that's the viability, 140 days. Actually, it's still growing. Actually, you have to use a special chamber to check the study, which is being approved by WHO. We have bought the chamber, and then we maintain, and then we have been monitoring. So far, it's beyond 140 days. It can stay at room temperature. But if you store at 4 degrees, it will stay for a longer time beyond that. So what my thing is, maybe if you send to a different continent, you can transport at the cold temperature, and once it delivered to that particular kampung or village or far away, I think it can still, you can use the room temperature.
Okay, Prof, we have 2 more questions. before we close the session.
Yeah.
So this question is from Mizanur Fakri. He says, thank you, Prof. Just out of curiosity, is it possible to recombine such established plasmid into a probiotic, in which also gives us simultaneous benefits?
Yes, yes. Actually, you can also use probiotics like lactobacillus. I mentioned to you, if I give the list of strains, you saw one is lactobacillus, it's a probiotic. And recently I found they're also using bifidobacterium as a strain. So basically, because you want to use the DNA vaccine for the delivery, so you should not integrate into the genome. So you should not do genome engineering, just construct a DNA plasmid, transform into the probiotic, and this probiotic should be able to deliver the antigen into the host cell. And the thing is that you make sure that that mammalian promoter which you put is working by transfecting on a cell line before you continue the study. So it will work. You can use probiotic as a vaccine, but some probiotics are not good colonizer, but it has a beneficial effect, but they are not good colonizer. So you should use a good colonizer. One more thing important is you need an adjuvant. Any vaccine you want to potentiate, you have to have a immunological adjuvant. Like injectable vaccine, they use alum and, remember the France adjuvant we use for animal immunization and so on. So this particular Vibrio, the beauty is it has a CTB. CTB is a very potent immunological adjuvant. It's built in. So it will, there are studies been published and they have used CTB as a immunological adjuvant. Even in the plasmid construct itself, they found that it gives a good several-fold increase in immunological reactions. So not only colonization, you have to use a good adjuvant. We don't want to have multiple doses. You just want to move on with that. Okay, next question.
Next question, Prof, is from Dr. Lim Yat Yuan. He says, hi, Prof, thanks for the talk. When would it be a good time to use RNA vaccine? In your knowledge and opinion, why would the vaccine against COVID-19 be using RNA vaccine and not break the cold chain?
I know there are many molecular biologists here. How many of you like to work with RNA? RNA is the one molecule which degrades very fast. Everywhere the RNA in our hand and bench, dead cells, RNase is ubiquitous. RNase is the enzyme that degrades. RNA is ubiquitous. So working with RNA, the stability, you have to keep them at cold chain. So you have to have a very strict cold chain to maintain the RNA. Would it be the good time to use RNA? I really don't know, but it is working in a short term. Long term effect, we really don't know. And what is the last?
The last question.
You really need a cold chain to do it. I think breaking the cold chain for RNA, I think someone develops a formulation. The easiest thing I can think of is you need to use an analog. Okay, you synthesize the RNA artificially, you put some analog, but the body might not react to it. Remember the antiviral we use, it can lead to toxicity and so on. So you have to balance the formulation very effectively. I think RNA vaccine still it needs a cold chain.
Prof, one last question. Are you all right?
Yeah, okay. I'll be happy to answer any question, no problem. It's actually a learning session, so no problem.
So this is from Siti. She asks, your vaccine is focused on 0139. Does it give protection against other Vibrio cholera strains?
We have not tested it. We have tested only against 0139. And you have to remember, but one of the major virulent factor for cholera is the cholera toxin. It neutralizes the cholera toxin, that I can say. When we inject in a model and we put a cholera toxin and put this, and the antibody produced by the animal model, it's able to neutralize the cholera toxin, that I can say. But our vaccine is focused only as 0139. I don't know why. We have been trying to develop a strain for 01. We tried... for some reason, we have developed it and we didn't proceed further. But however, we found this, it has a ubiquitous use. It can be used as a vector for many other diseases.
All right, that's all the questions that we have today.
Okay, just letting you all.
We thank you very much for Prof. Ravichandran for spending your time and giving us a very insightful talk today. The MSMBB and Department of Parasitology would like to thank everyone for spending your time with us. With that, let's close this session. Thank you and have a nice day, everyone.
Thank you. Have a nice day.
Thank you. Thanks for Ravi.
Thank
Thanks.
you.
Bye-bye.
Bye.